Hello, csir aspirants. This is a continuation of Immunology series, and we are back again with another video. And today we are going to study the antibody diversity and the hypersensitivity. So, these two topics that we are going to cover in this video. Okay. Antibody diversity, right? Antibody diversity basically. Diversity in antibody is generated by somatic cell recombination and DNA rearrangement occurring in activated B cells. So basically, whatever the somatic recombination and DNA rearrangements happens in activated B cells, due to that the diversity in antibody is generated. Okay, so variable regions of heavy chain and light chain is polygenic in nature. See, basically this is antibody, right? This is light chain, this is heavy chain, right? So in light chain, it is encoded by V and J chain. V stands for variable and J stands for junction. Whereas in heavy chain, it is encoded by V, D and J genes. V for variable, D for diversity and J for junctions genes. Okay, so basically light chain and heavy chain. Light chain, this is the exon of light chain. The C terminal is contain lambda 1, lambda 2 and kappa, and in the ln stand the ln thing, like this kind of stuff, there is a variable thing and junction thing, and the nucleotide difference between the variable and junction is 40 and 3. Okay, same as for heavy chain, there are basically three exons are present, like variable, diversity and junction, and the constant part, like this part. The constant part is divided into different aspects due to some somatic combination, DNA rearrangement. So different type of antibody is generated by from the constant region. So the series and the pattern they follow is the first IgM form, then IgD, like M, D, gamma 3, gamma 1, then A, then G2, G4, E, A2. So this is the pattern that diversity follow. What happens basically, there is a site where random mutation or recombination happened by the help of VDJ recombinase or RAG, which is a recombination activating genes. And here the two as when we compare with the heavy chain, there are the two two site where the site of recombination happen by the help of RAG1 and RAG2, which is again the product of recombination activating genes. Two important theories are related to antibody diversity. First is germline theory and second is somatic cell recombination theory. Germline theory, it states that like antibody encoded by large number of different genes which recognize specific antigen, and this theory was discarded because number of antigen always greater than the number of limited genes. So germline theory is not applicable. Somatic cell recombination theory, what they are saying, they state that number of antibody producing genes are limited and diversity is generated by DNA rearrangement and somatic cell recombination. That is very true, right? Here they were saying that number of antigen are always greater than, no, basically, and then the limited genes. Basically the number of antibody producing genes, they are limited. So that is why the diversity needed. By the DNA rearrangement and somatic cell recombination. So the previously that we have seen in the diagrammatic form, there it is written in a theory form that like in the light chain recombination occurs between variable and junction exon, whereas in heavy chain recombination occurs between the variable, diversity exon and D and J exon. VDJ recombination and RAG1 RAG2 is responsible for causing double strand break and generating diversity in antibody. So random nucleotides are incorporated at the site of recombination. So they are also known N nucleotide, and which are incorporated template independent manner. Okay. So in T cell receptor recombination occurs in the variable region of alpha and beta polypeptide chain. Multiple VJC are responsible for producing T cell receptor. So somatic cell hypermutation do not occur in the T cells, and RAG1 and 2 knockout mice. If RAG1 and RAG2 they are knockout, then The mice are immune compromised cells because the antibody diversity is less, so that is why. So different process occurring for antibody diversities are combinational joining of VD, J and C, association of different heavy chain with light chain, somatic cell recombination of VD, J recombinase, somatic cell hypermutation, different folding of antibodies like P addition that has happened, N addition. So these are the methods, for because of that the antibody diversity happen. So what is junctional diversity? Basically, the precise position at which the genes for V, variable and junctional or the VDJ segments are fused together and is not fixed. And imprecise DNA recombination can lead to change to amino acid, and these junction sites called junction diversity. So basically, there is the change in amino acids, and that is why the diversity happen. Now the second topic is hypersensitivity. We are complete with antibody diversity. So hypersensitivity, there are four types of hypersensitivity: type 1, type 2, type 3, type 4. So let's begin with type 1 hypersensitivity. So basically it is caused by allergen, which is also called anaphylactic reaction. So IgE provide immune response, like in this mast cell, basophil, eosinophil participate in this response. Okay, if there is an allergen, then the mast cell, basophils and eosinophil will participate. Allergens may be extracellular molecules like pollen grain or food components like antibiotics and albumin, etc. So in this there is a hyperactivation of mast cells occur. So most common allergy is asthma. Asthma is an allergy. Inhibitor of allergy include epinephrine and green tea. And if we could take epinephrine on green tea, it is normally an inhibitor of allergy. So type 1 skin test is performed in which allergen is introduced intradermally, like a skin on skin. So hypersensitive response generate wheals and flares. Basically in type 1 the symptoms that happens is red patches or burning sensation, which we could say like a wheal and flares. Okay, now so basically these all are primary immune response. Let's see about the type 2 hypersensitivity. In type 2, it is basically mediated by IgG and IgM antibodies. In this, it is mediated by IgE antibody. Okay, so it is caused by Irish factor, which causes erythroblastosis fetalis. Erythroblastosis fetalis. What happens in this? Basically, Irish negative mother react with second child if that child is Rh positive. So basically it has the antibody against the Rh positive. So that is why erythroblast fetalis happen. It is also caused by incompatible blood transfusion, which can lead to anemia. So basically antibody directed against cell surface and antigen mediated cell destruction. So via cell destruction via complement system by creating pore in plasma. The second method that can occur by antibody dependent cell mediated cytotoxicity, that is ADCC in NK cell. Opsinin, basically opsinin go in the macrophages, and due to that the phagocytosis happens. Type 3 hypersensitivity. Type 2 is all about this blood incompatibility. Type 3 hypersensitivity, in this immune complex deposition occur in the body, and why it can lead to dilation of blood vessels, kidney failure, osteoarthritis, like a joint break. It is mainly caused due to the presence of large accumulation of non-degradable antigen and antibody, that is IgG and IgM again happens in this. So what happen in type 3, basically autoimmune disease, whatever the autoimmune happen, this due to the type 3 hypersensitivity. Okay, for example, SLE, that is the systemic lupus erythematosus, and this immune complex with many organ, like in this the butter syndromes normally happens, like in the chicks there is a patches and it appears like a butterfly syndrome. So what happened in this, basically antigen or antibody reaction happen and activate the complement system. And basically in complement system, particularly C3b can bind with the antibody. Okay, and and the C3a part will activate other cells, and they just go and come out with that they just go and due to that, many other cells come and at that place as a chemokinin, and the reactions happen. Okay. The other example is vasculititis. In this, immune complex bind with the blood vessels. The third example is glomerulitis, and this immune complex bind with the kidney. Now, what is type 4 hypersensitivity? Type 4 hypersensitivity, in this there is a no, they basically antibody doesn't play any role. It is a delayed type of hypersensitivity. What happened in this, like normally antigen presenting cells like MHC2, they present the antigen to the naive T cells, and after naive T cells, with the help of CD4, it will going to activate Th1 cell form. And Th1 particularly we already seen about the in the previous video. Hope you will see the previous videos of immunology. Th1 will activate the macrophages, and after activation it is caused by the by mycobacteria. Now, and does the granuloma formation occur? That is the number of many macrophages activated. Now, the poison case is also the type 4 hypersensitivity. Okay, it is a cell mediated, delayed, and antibody dependent, independent hypersensitivity. So yeah. Let's see a tabulate form of the whole hypersensitivity topic. The reaction description, participating antibody, skin reaction, typical time of onset. So in the CS exam, there are few questions which arise from the hypersensitivities. So we should remember like which kind of antibody is required and which kind of hypersensitivity. So in type 1 description, it is an immediate type of hypersensitivity. For example, allergy. It is immediate, okay? So, and the participating antibodies are IgE plus mast cells. The skin reaction is allergy skin test we can do. It is immediate, so there is no time onset. Type 2, antibody to cell associated antigen. Example, hemolytic anemia. In this, IgG and IgM antibody participating. Type 3, antibody to antigens solution. Immune complexes basically happens, and in this serum sickness or Arthus reaction hota, and it requires IgG and IgM again. So the typical time of onset is 7 to 10 hours. In type 2 hypersensitivity, delayed type hypersensitivity, T helper cells, like example graft rejection. The test we can perform is tuberculin test, and the typical time onset is 1 to 3 days. So this is all about hypersensitivity and antibody diversity. I hope these video help you in preparation, and as you can know that the exam can happen in April, so we have very less time to prepare. So yeah, till that, happy learning, all the best.